Good morning, sir.
Okay, good morning everybody. Now 10:05. I think we should get start. Am I audible to all?
Yes, sir.
Okay, I believe it is visible.
Yes, sir.
I would like to review the last day's lecture, whatever we have discussed. First, let me review once and then we'll proceed to the next. Okay. So last day, the important portion that we have tried to cover, that is the cell wall structure of the bacteria. And as we have mentioned that bacterial cell wall is one of the most important component which basically provides the particular shape, size, and most importantly, the protection or defense for the bacterial cell. Because of this peptidoglycan, I mean the cell wall, bacteria remains very much stable. They could protect themselves against different kind of adverse environmental conditions. Cell wall, basically, as we have mentioned, that this is the cell wall of the bacteria. It is made up of one very unique chemical component that is known as peptidoglycan or murin, which is unique in this earth. It is available only in case of the bacteria. No other living cells, it does contain this peptidoglycan in their cell. Nowhere, in any part of the cell, neither in eukaryotes nor in the archaea, only prokaryotes, the bacteria, they do carry this peptidoglycan in their cell. And it is basically made up of two sugars, alternate backbone of these sugars, N-acetyl glucosamine and N-acetyl muramic acid, alternating chain. They duly made a chain, and multiple chains, they will be linked by the peptide chains. And those peptides, first N-acetyl muramic acid, it is linked with 4 amino acids, alternatively D and L form, and same available in all kind of N-acetyl muramic acid. Then both the chains, they do linked with a pentapeptide, 5 amino acid, that is the L-lysine, L-glycine, 5 molecules, they do link with the third amino acid of the first chain, goes to the fourth amino acid of the next chain. By that way, it makes one very strong network of the sugar and amino acid. So this is the entire structure of the peptidoglycan. This kind of linkage, this kind of layer, it is multiple layers, they do form the cell wall structure of the bacteria. Then the cell wall, as last time we have talked about, it is having multiple functions. They offers variety of activities for the bacteria, like they do counter the osmotic pressure, they helps in the attachment of bacteriophage. It also provides the appearance or space for the flagella, fimbriae, pili. They are also cell divisional advantage. They are protecting bacteria against antibiotic, as well as they are also one of the part of the antigenic determinants in case of the bacteria. So altogether, in a nutshell, if we say that cell wall of the bacteria, it is one of the most important component for the bacterial structure, which provides the defense and also maintains the shape, size, particular shape and size. Now, when we talk about the bacterial cell wall, so far we talked about only the component that is known as the peptidoglycan. Peptidoglycan, okay. Now, bacterial cell wall, as so far we have already discussed, it is not that much of simple whatever we have talked right now. We talked that bacterial cell wall is composed of the peptidoglycan, but that is not the end. Basically, based upon their structural difference, we can divide the bacteria into two groups. We have mentioned bacteria into two groups. One group is known as gram-positive bacteria. Another group is known as gram-negative bacteria. Gram-positive and gram-negative. If you remember back, we have in the history of microbiology chapter, we have mentioned the name of one gentleman. He is the Alan Christian Graham. Alan Christian Graham, that gentleman, he has developed one technique for staining of the bacteria, how to demonstrate the bacteria under microscope. For that, he has developed one staining technique. By that staining technique, we can observe the bacteria under microscope two different way. One group, it is looking like that purple color. Another group is looking like a bright pink color. Intentionally, I wrote the gram positive in purple, gram negative in pink, just to indicate that under microscope, how would they look like. So gram positive, the bacteria by gram staining. Gram staining means it is a method of staining by which bacteria can be observed under microscope. One method of staining by which bacteria can be observed under microscope. This technique is developed or it identified by Alan Christian Graham. After his name, this technique or name is known as gram staining. And accordingly, bacteria, two type of bacteria we can see. One gives a color like purple color. Another bacteria gives it very intense pink color. So the purple colored bacteria, this is known as gram positive, and the pink colored bacteria is known as gram negative. Okay, so based upon but this differentiation, color difference, it is because of the bacterial cell structure. Underline it, you can note it down. The coloration, it appears sometime purple, it appears sometime pink. The difference appears due to the structural variation of the cell wall of the bacteria. So what is that? That is the topic of that now. Here, I have given two pictures. First, we look at the picture at the bottom. Picture at the bottom. The picture at the bottom, it indicates the difference of two group of bacteria. One is gram positive, another is gram negative. Here, this is the internal structure, internal component, the cytoplasm of the bacteria. It is already marked. First layer beyond cytoplasm, it is the cell membrane, or we say cytoplasmic membrane. Both the cells, gram positive, gram negative, we can say this is the cell membrane, same size. Then the thick purple layer, a thin purple layer, it is here. This is the peptidoglycan. Just now we have discussed. So this is the peptidoglycan. But in this group of gram-negative bacteria, if you see, after peptidoglycan, one more layer is available. This layer is known as outer membrane. Please note it down. This layer is known as outer membrane, outer, O-U-T-E-R, outer membrane. So the basic difference between the cell wall of the gram-positive bacteria and gram-negative bacteria is that in case of the gram-negative bacteria, they have an extra layer in their cell wall. An extra layer in their cell wall, that is the external, extremely outside. This layer is known as outer membrane. This outer membrane is absent in case of gram-positive bacteria. Okay, now look at the thickness of the peptidoglycan. In case of gram-positive, the thickness is much more, obviously, whereas the peptidoglycan layer, the thickness in case of gram-negative bacteria, it is less. So these are the two basic visible observable difference in case of the cell wall of gram-positive bacteria and gram-negative bacteria. Let me repeat. This is very important part in the structure of the bacteria. You should understand it, that in bacteria, we can divide into two groups based upon their cell wall structure. They are known as gram-positive bacteria and gram-negative bacteria. So how do they look like? Gram-positive bacteria, by gram staining, they looked purple colored. Gram-negative bacteria, by gram staining, they look intense or bright pink color. Now, if we look in the inside of the bacteria, the ultrastructure of the bacterial cell wall, then we can find the gram-negative bacterial cell wall is little bit complex compared to gram-positive bacteria. What type of complexity it is? That gram-negative bacterial cell wall, it contains an extra layer outside their cell wall, that is known as outer membrane. Outer membrane. Then the peptidoglycan layer in gram-positive bacteria is thicker compared to the gram-negative bacteria. Okay, everyone? Understandable?
Yes, sir.
Yes, sir.
Okay. Now we need to look little bit in details about this outer membrane or the gram-negative bacterial cell wall. So far we read about the peptidoglycan. Okay, here is the gram-positive bacteria. We have made it a little bit bigger size. This inside this layer is cell membrane. We'll talk later on what is about the cell membrane. This is the peptidoglycan. We can see that layers, multiple layers made, right? And this is a very thick layer, peptidoglycans, and outside there are certain more molecules. It is read as a teichoic acid. I'll discuss later on. Teichoic acid, it is available in case of this gram-positive bacteria. But if you look at the gram-negative bacteria here, this is the cell membrane. This is the peptidoglycan. You can see it is much thin layer. Then outside, this is the outer membrane. This is the outer membrane. Outer membrane, it is very complex structure. Very complex structure. Mostly, it is made up of phospholipid and protein. Outer membrane, it is mostly made up of the phospholipids and protein. And it is having 11 to 12% of the lipid, 11 to 12% lipid. That's why if we compare the cell wall structure of gram-positive and gram-negative, the lipid content will be much more in gram-negative bacterial cell wall than gram-positive. We should note it down, all those points. The lipid content of the cell wall of gram-negative bacteria will be more compared to the gram-positive bacteria. This is because of the outer membrane, because the outer membrane is mostly made up of phospholipid and protein. Okay, this outer membrane, now I will try to show you a little bit bigger picture. Here, look at this picture. Here is the gram-negative bacteria. Once again, at the bottom layer is cell membrane. Then this is the layer peptidoglycan. Then this is the outer membrane. So this is a layer. Here it is. This is the cell membrane. Then the small thin plate is the peptidoglycan. Then this layer is the outer membrane. Okay. This outer membrane, basically, it is, as I told you, composed of the phospholipid. It is having three important components. Outer membrane, it is composed of three important components. One, it is known as lipid A. Write it down. This yellow box, can you see? Yellow boxes, this is the lipid A. Lipid A. Second component, this is known as core polysaccharide. This is basically the core polysaccharide. And then third one, it is the O somatic antigen or O polysaccharide. Once again, please note it down. The outer membrane is composed of three major components. One is the lipid A, another is core polysaccharide, another is O polysaccharide. So altogether, this is a phospholipid membrane, thick phospholipid membrane. This is phospholipid membrane. It is known as PLP, protein lipid protein membrane. Protein lipid protein membrane that is embedded with three important components: lipid A, core polysaccharide, and O polysaccharide. This O polysaccharide is also known as somatic antigen or O antigen. Somatic antigen or O antigen. O, capital letter O. Somatic antigen or O antigen. Lipid A, core polysaccharide, and O antigen or somatic antigen. This is the entire component of the external lipid A, O polysaccharide, core polysaccharide of the outer membrane. This is altogether also known as endotoxin. Note it down. This is altogether known as endotoxin. Okay. Now, one more terminology I will add. This is known as LPS, lipopolysaccharide. Lipopolysaccharide. Let me repeat the gram-negative cell wall again. What I have said, this is an additional membrane available in case of gram-negative bacteria beyond the peptidoglycan. Fine. This is mostly made up of the phospholipid layer. This is the phospholipid layer. Here you can find it out. Then it is having LPS, lipopolysaccharide. The lipopolysaccharide has three components. One is lipid A, one is core polysaccharide, one is somatic antigen. Phospholipid membrane and LPS or lipopolysaccharide. Lipopolysaccharide has three components. So lipid A, core polysaccharide, somatic antigen. Everyone, this lipopolysaccharide or LPS, this is also known as endotoxin, endotoxin of bacteria. Lipopolysaccharide is also known as endotoxin of bacteria. Now, if question asked to you that gram positive or gram negative bacteria, which one can produce endotoxin? So answer is obviously the gram negative bacteria, because endotoxin means it is a part of the outer membrane, and outer membrane is present only in case of gram negative bacteria. So endotoxin, it is the symbol for gram negative bacteria. Gram positive bacteria do not have endotoxin. Do not have endotoxin. One more important part I should mention here. This is an extra layer. Now I am going back to the picture. This is an extra layer. I said it is outer membrane. This is peptidoglycan. Now, as this is an extra layer, it should have attached, the outer membrane should be attached with the peptidoglycan, okay, so that it should not be removed, it should not go out. The peptidoglycan layer and the LPA outer membrane, they used to bind with one protein. Can you see this red colored protein? They are anchoring protein, means they are holding the outer membrane hardly bound with the LPA peptidoglycan. These proteins is known as bronze lipoprotein. Bronze lipoprotein, if it is written somewhere, the picture, let me see, bronze lipoprotein should be written somewhere. If not, then you can write it back. Okay, you can write it back. B, capital B, B-R-A-U-N, B-R-A-U-N, apostrophe S, bronze lipoprotein. This is the anchoring protein which holds the outer membrane attached with the peptidoglycan layer in case of gram positive bacteria. This is the bronze lipoprotein, or it is also sometimes known as only lipoprotein, which is binding the or holding the outer membrane with the peptidoglycan. One more terminology we need to add here, that when there are multiple layers, look at that. This is the cell membrane, this is the peptidoglycan, this is the outer membrane. So obviously, what will happen? There will be some blank space between the layers. There will be blank space between the layer. So those blank space, this is known as periplasmic space. This is known as periplasmic space, okay? The space between the multiple layers of the cell wall. We have the cell membrane, we have the peptidoglycan, we have the outer membrane. Obviously, as there are the different membranes layered one by one, so there should be some gap in between. So those gaps, this is known as periplasmic space. Now, I have a question to all of you, that which bacteria will be having more or the bigger periplasmic space, gram positive or gram negative bacteria? Shakib Sarkar, can you answer?
So gram positive.
Okay, actually answer should be exactly opposite. it should be the gram-negative bacteria, because you see there are multiple layers. Let me show picture here. Okay, this picture. This is the space between two, and here this is the cell membrane, and here is the outer membrane. Can you see that which one is having more space in between? Obviously gram-negative. Here one layer, here one layer. Okay, so gram-negative bacteria, they have more periplasmic space. Space means this is one, this is layered. But in gram-positive, you can find only one layer between the cell membrane and the cell wall. Is it okay, everyone?
Okay, sir.
So now what we have made, we are mentioning different differential points between gram-positive, gram-negative. If all of you have listening concentratedly with concentration, you can find many points we are raising gradually. So these will be enlisted as the differential points for between the gram-positive bacterial cell wall and gram-negative bacterial cell wall. So we have mentioned that we have started with the gram-positive, gram-negative. That gram-positive bacteria, peptidoglycan layer is thick, gram-negative, thin layer. Gram-negative bacteria has an extra outer membrane. In gram-positive, it is absent. Is it okay? Then the endotoxin is available in case of gram-negative bacteria, absent in gram-positive bacteria. Then the periplasmic space is thicker in gram-negative bacteria, thinner in gram-positive bacteria. One more point, teichoic acid, although I have to talk in next few slides, teichoic acid is available in gram-positive bacteria, absent in gram-negative bacteria. All right. So these many important points you should mention and you should note it down, which will make the difference between the gram-positive and gram-negative bacteria. Now, once again, let me go back to this. There are one more part. Here, look at this. Can you see different kind of big size molecules? It is drawn here, right? One is this brown colored, thick, big size. This is a protein. This is the hollow protein. These are all protein molecules. What is happening? These proteins are transmembrane protein. Transmembrane protein. Now look at the terminology. Transmembrane means a protein which can pass throughout the thickness of the membrane. Means this is the thickness of the membrane. It passes from one end to another end. So that's why it is transmembrane protein. So there are many protein, transmembrane protein, through which the molecules, including water molecules, can pass through the membrane. Otherwise, this layer is impermeable. The layer outer membrane, it is impermeable membrane, means nothing can move through it. Neither water nor air, nothing. But bacteria cannot survive without the movement of the water or air molecule. They need the respiration, they need the water, they need food, they need to excrete the excreta, obviously. So altogether, there should be some channels. Those channels are such protein, which are transmembrane protein. This kind of protein is known as porine protein. Note it down, P-O-R-I-N, porine. Porine protein means these are the transmembrane protein which helps in the movement of solute or solvent, means it may be anything, through the impermeable membrane. Through the impermeable membrane. Such kind of protein is known as porine protein. Okay, everyone? Anyone wants to raise any doubts about whatever we have talked so far? Anyone? Okay. I think I am audible to all.
Yes, sir.
Fine.
Yes, sir.
So it indicates that I could make it a little bit clear to all of you. So we have talked about the bacterial cell wall. Bacterial cell wall is composed of major component is peptidoglycan.
Peptidoglycan.
Then another component also available, that is the outer membrane. But outer membrane is available only in case of the gram-negative bacteria. In case of outer membrane, there are two components. One is a phospholipid bilayer, another is the LPS. LPS is also known as endotoxin. The LPS has three components. One is lipid A, core polysaccharide, and somatic antigen. Okay, this is the outer membrane. The outer membrane is linked with the peptidoglycan with a protein that is known as bronze lipoprotein. Then the space between the outer membrane and cell membrane of gram-negative bacteria, it is known as periplasmic space. The space between the peptidoglycan and cell membrane in gram-positive bacteria, it is the periplasmic space. Periplasmic space is much bigger or wider in case of gram-negative bacteria than gram-positive bacteria. Now, you should ask one question, that what is the utility of this periplasmic space? The periplasmic space is basically bacterial storehouse. It is a storage place. Here, bacteria can keep many things for their storage. It may be the excreta, it may be the food for future. So it is a storehouse for the bacteria. They maintain all the molecules as the storage in the periplasmic space. So the protein molecules available in the periplasmic space, they are known as periplasmic protein. If any protein is available in the periplasmic space, they are periplasmic protein. All right, so this is about the cell wall. Now let me understand that what about the LPS, lipopolysaccharide. Lipopolysaccharide, as I said, it is also known as endotoxin or a pyrogen. Anyone who wants to speak, what is pyrogen? Did you anyone heard about so far? What is pyrogen? If yes, you can talk. If no, no issue. I think you have not heard so far. Pyrogen means something which, yes, please.
So which produce fever?
Yeah, correct. Very good. Which produce fever. Absolutely correct. So pyrogen means any agent, chemical or biological agent, which produces fever in the living body, particularly the human animals. So it is a pyrogenic agent, means endotoxin can generate fever. All right. Then, as I said, structurally it is composed of lipid A, core polysaccharide, O antigen, already we have talked about. What about their property? They are toxic. Endotoxin, the LPS, it is a toxic molecule. It can kill mice, pig, human. Yes, it is correct. Whenever, if anyone inject pyrogen or endotoxin in excess amount, that can kill the living object. Or even, this pyrogen is available in most of the cases. Whenever we do suffer from any infection of gram-negative bacteria, generally people feel fever or animals feel fever. That fever is mostly due to the pyrogen, means endotoxin. Gram-negative bacteria, they associate with septicemia, and that death is due to LPS. The same line I talked like that. If any individual, any animal, they do suffer from septicemia. Septicemia means when the blood of animal or human, it contains bacteria as well as toxin. Bacteria as well as toxin, they are known as septicemia. So that septicemia, if the individual dies, it is due to the LPS. DPT, as an example, I think all of you know the DPT. What is the full form of DPT? Who will say? DPT, it is a term of vaccine. All of us, we have taken
Diphtheria,
this
sir.
vaccine. Diphtheria, D for diphtheria. Good. Next, Rahul talked like diphtheria. Who will talk more?
Pertussis,
Yeah,
tetanus.
pertussis, very good. And T for tetanus.
Tetanus.
Great. Very nice. That means we took the vaccine against 3 different diseases as our very early stage of our life, mostly 3 months, starting from 3 months. Diphtheria, pertussis, tetanus. And it is true, whenever we do inject the DPT to any kid, it is obvious that the kid will develop fever. So that fever, it is due to the presence of the endotoxin that we inject through the bacteria. And it says that if the kid does not develop the fever after giving DPT, you can raise a question that there might be something wrong with the vaccine. it should develop fever, and that fever is good. Then this LPS, it also used as an adjuvant. The term adjuvant means something which can stimulate the immunity. Term adjuvant means something which can stimulate our immunity. Now, I think in today's date, due to this COVID-19 pandemic, all the group of people learned that what is the immunity booster. Will say that please improve your immunity. Many people has the many advice, take this food, that food, do that like that way, so that you can raise or improve your immunity. So that is a stimulation, means you are stimulating, just like the exercise. We do some of the workout exercise so that our body will be strong. So you are helping our immune cells to remain healthy, stimulated. So that is the immunity booster. So adjuvant is the something which actually helps in the improve or stimulate the immune status of the body. Then this LPS, they are heat resistant, very hard to remove. LPS is highly heat resistant, means if you boil water, if it contains LPS, you cannot remove it. It is very tough to remove. Suppose sometime we are using the water, that water also contains LPS, but it might be very mild quantity. We are using the injection, injectable water. Sometime we do go to the pharmacy. If you need an injection, we do need to purchase the powdered antibiotic or powdered medicine, and that should be dissolved in the water. But that water cannot be the tap water or something from our filter. You need to purchase it from the pharmacy. It is available in an ampoule that might be 1 ml, 2 ml, 3 ml. What is the important specificity of that water? If you read the level of that water, it is written pyrogen free. It is written the pyrogen free. Pyrogen free water is important because if you are injecting pyrogen along with all different medicine, means you are actually developing fever in the body of the patient. Sometimes it is very much lethal. We cannot do. So pyrogen free water is very important, and it is not easy. Even if we boil water, pyrogen will not be removed. So the LPS or the endotoxin is very heat tolerant, or it can heat tolerant, means it can tolerate the heat. So these are the properties of the LPS or endotoxin. Okay. Now, one more terminology here I would like to mention. One is known as protoplast, one is known as spheroplast. For that, I need to go back to the previous picture. Here, look at this picture once again. This picture once again, we have given the picture of gram-positive bacteria and gram-negative bacteria, right? Gram-positive bacteria, internal structure is a cell membrane. Then gram-positive, one layer of peptidoglycan, gram-negative peptidoglycan, thin layer. But the extra layer is what is this? Outer membrane, made up of the phospholipid or protein bilayer. Sometimes some of the agent, say, let me take an example of enzyme lysozyme. I think many of you might have heard about the enzyme lysozyme. Lysozyme is an enzyme that is available mostly in many areas, particularly the egg white. Even in our buccal cavity, through our saliva, we do secrete lysozyme enzyme. This lysozyme, what does it do? It can selectively lyse or destroy the peptidoglycan. An enzyme lysozyme means if you have the lysozyme enzyme with you, and if you are giving it to the gram-positive bacteria, then its cell wall will be destroyed. Okay. Same thing also in case of the gram-negative. Cell wall means which part? Peptidoglycan. Then what will happen? The gram-positive bacteria will lose its cell wall. What will be the available part? Only it will be covered by a cell membrane. Let me repeat. We have the gram-positive bacteria, and if we treat it with an enzyme that is known as lysozyme, then lysozyme can destroy the layer of peptidoglycan. So peptidoglycan will be removed. Then what will be available? Only left part is the cell membrane. Look at the gram-negative bacteria. Gram-negative bacteria also, if you treat with lysozyme, then what will happen? The peptidoglycan will be removed, but still it will be having two layers, one cell membrane, one outer membrane. Is it okay, everyone? Now, by that way, here is the gram-negative bacteria, here is the gram-positive bacteria. Gram-positive bacteria, as I said, cell membrane followed by peptidoglycan. Gram-negative bacteria, cell membrane followed by peptidoglycan, then outer membrane. When both of them treated with certain chemical, then what will happen? Gram-positive bacteria will be converted into this. They will be having only a layer of cell membrane. Okay, they will be having a layer of cell membrane. This structure is known as protoplast. Gram-negative bacteria, if you treat them, once again peptidoglycan will be removed, but it will be covered with two layers, one cell membrane, one outer membrane. This is known as spheroplast. Now, one more important point, look at that they were oval shaped or rod shaped. After making it protoplast or spheroplast, both are circular. Can anyone explain it? Why? Is there anyone?
Because cell
Yes.
wall will decide the shape.
Absolutely correct. The cell wall means peptidoglycan. I have repeatedly told that the peptidoglycan is the hardest structure that maintains the shape of the bacteria. When peptidoglycan removed, bacteria lost its original shape, because this is the best structure to maintain the buoyancy. So all bacteria, if it loses its peptidoglycan, it will become spherical. So protoplast is the modified form of gram-positive bacteria when their peptidoglycan lost because of certain physical or chemical agent which removes the peptidoglycan. Then it forms the structure, it is known as protoplast. So protoplast means cytoplasm covered by cell membrane. Gram-negative bacteria, when you treat with that same treatment, then they will make a spheroplast. The basic difference is that spheroplast will be covered with two membranes, one cell membrane, one outer membrane. But this is also spherical because the peptidoglycan lost. All right, both, all of you. So this is the protoplast and spheroplast. But one line, please mention that both the protoplast and spheroplast, these two form of bacteria, they are highly unstable. They cannot survive for a long time because this is very, very fragile membrane. It can be ruptured at any time. Once again, you can remember or compare with the egg cell. When you have the very hard egg, don't boil the egg. Try to remove the hard calcium egg cell. Then if you can maintain that shape of the egg, no, because the inner cell membrane is very, very thin and highly fragile. So it will be lost at any time. So the same structure is the protoplast and the spheroplast. Highly fragile, cannot maintain its fragile in the buoyancy, cannot maintain its structural definition. Okay, so altogether we have completed the cell wall structure. Now I'll go to the teichoic acid. I have mentioned it. Teichoic acid, it is present only in case of gram-positive bacteria. It is absent in gram-negative bacteria. Teichoic acid is mostly composed of glycerol, phosphate, and ribitol. It is also termed like that ribitol phosphate. Okay, glycerol and ribitol phosphate. But the teichoic acid, basically, what is its exact function, that may not be very much clear, but sometimes it is observed that they help in the attachment of bacteriophage on the surface of the bacteria, gram-positive bacteria. So these are the teichoic acid molecules. Some of the teichoic acid are transmembrane. It is known as lipoteichoic acid. Some of the teichoic acid which is non-trans, only very small part, this is only teichoic acid. Those which are transmembrane passing through the entire peptidoglycan, then it is bound with cell membrane, they are lipoteichoic acid. You can see. So now another question was that who binds peptidoglycan with cell membrane? Here is that lipoteichoic acid. Look at that. It binds with the cell membrane, and this is the peptidoglycan. So lipoteichoic acid is the anchoring agent which binds the our peptidoglycan with cell membrane of the bacteria. Same for gram-positive, same for gram-negative. In case of outer membrane, it bounds with the our peptidoglycan, that is the bronze lipoprotein, and here it is the lipoteichoic acid. But teichoic acid is unique for gram-positive bacteria. It is absent in gram-negative bacteria. Another structure in the bacterial cell, this is known as a mesosome. Mesosome means this is an ultrastructure of the bacteria. Now look at this is the line I am passing through the cursor. This is the cell membrane of the bacteria. And external to that thick layer is the cell wall. This is cell membrane. The same cell membrane, sometimes it becomes a fold and then it embedded into the cytoplasm and it makes one structure inside the bacterial cytoplasm. This kind of structure, it is known as mesosome. So it is written that mesosomes are specialized structures that formed by convoluted invagination of cytoplasmic membrane. Look at that. Same membrane, it invaded into the cytoplasm. It is not specialized, unique and separate entity. It is the same membrane, makes a fold and gets inside the cytoplasm. Then it is the mesosome. So sometimes there are two types of mesosomes available. One is the septal mesosome, another is the lateral mesosome. Septal mesosome means it can make a septation, means division of the bacteria in 1 to 2. And those are helping the bacteria to divide. And the lateral mesosome is the very small mesosome that does not have any role in the cell division. So this much of the function are known so far, what is about the mesosome. But this is very commonly available structure of the bacterial cell, means presence of the mesosome. It is sometimes there. Next component, let me talk about the cytoplasm of the bacteria. Cytoplasm of the bacteria, obviously, this is the house for all the internal organelles. All of you, please remember to mention the terminology. When we talk about the eukaryotic cell, we know the eukaryotic cell contains different of cellular organs like Golgi bodies. Then they are having the plastid, means chloroplast or leucoplast. There might be having mitochondria. There will be nucleus. But in case of the prokaryote, means bacteria, almost absent. No cytoplasm, nucleus, where the nucleic acid or the nucleic acid, that is the DNA, it remains floating as a nucleoid or nuclear material, no nuclear membrane. So let us talk about the cytoplasmic content. 80% of the cytoplasm is actually water, 20% are salt and proteins. Here, maintenance of the osmotic pressure of the cellular content, that maintains by the cytoplasm. When the organism, they sustain either in isotonic, hypertonic or hypotonic environment, accordingly the cytoplasmic content varies. When the bacteria sustain in the hypertonic environment, at that time the solute content will be more in the cytoplasm. because just to maintain the osmotic pressure inside and outside. Similarly, when bacteria survives in the hypotonic environment, this solute content will be less. So this 20% content may be variable. This is all about the isotonic environment. In case of the bacteria, it is circular and haploid. Please mind it. It is circular always. Bacterial nucleic acid is always circular and is haploid in nature. The advantage of the haploid means 1N DNA over 2N, because it is very efficient, can multiply very quickly, and can adapt with the environment very quickly. If you can look at the evolutionary changes or adaptation of eukaryote and prokaryote, prokaryotes are rapid. That's why the viruses, just like our coronaviruses, if you look, that every 2 months probably they are mutating themselves. Matter is so simple. Nucleic acid is so simplified nucleic acid, it can edit and it can mutate it like anything. But same mutation is very, very difficult in case of the eukaryotes and particularly the higher level of the animal or plants. Our mutation, it takes hundreds of years. But in case of the smaller object, the prokaryotes, where the nucleic acid is haploid type, they can mutate easily. So they can adapt to the environment immediately. So whether our earth temperature goes 55 degrees centigrade average, bacteria will not be having any problem. But probably we will extinct from this earth. That is all about because of our nucleic acid configuration. Cytoplasm of the bacteria also contains additional or extra chromosomal DNA. It is known as plasmids. That is the extra chromosomal DNA, the plasmid. We will talk about plasmids, of course. That helps in the antibiotic resistance, and that also help in the transfer of genetic property from one bacteria to another. There are no organelles like mitochondria or Golgi body available in the cytoplasm. So it is a very simplified form of content, the bacterial cytoplasm, compared to the eukaryotic cells. Plasmids. This should be the last topic of the day. Plasmids. I believe that all of you heard about the terminology of plasmid. In our body, in our cell, we have the nucleic acid. That is also, of course, one double-stranded DNA, linear. Am I right? We have a very complicated nucleic acid, a DNA, that is always double-stranded. That is a linear, and that makes the chromosome. Then it makes the complete setup of our nucleic acid. But in case of the bacteria, they have two types of nucleic acids. Actually, three types: the DNA, the RNA, and then extra DNA. That is a plasmid. RNA and DNA is there, but in case of the DNA also, something extra is available. So this is the bacterial DNA, that is the chromosomal DNA, but it is having one extra DNA, that is the plasmid DNA. Plasmid DNA are very much unique and different. They are always circular, double-stranded, high supercoiled. Let me make it once again. They are available in the circular, double-stranded, supercoiled, self-replicating agent. Self-replicating agent means they are not dependent on the bacterial DNA for their replication. So the plasmid DNA, it is having the capability of self-replication, contains genes that confer some property such as antibiotic resistance and virulence factors. So they can carry the certain different kind of factors. Plasmids are not essential for cellular survival. If any bacteria carries plasmid, it is an added advantage. If some bacteria do not have a plasmid, that does not mean it will not survive. It can survive easily. So it is not a survival gene or survival nucleic acid. If bacteria is independent of plasmid, it may have, may not have. There are three different types of plasmids available in case of the bacteria. One is known as F plasmid or fertility plasmid. Another is known as R plasmid or resistance plasmid. Another is known as coal plasmid or colicin plasmid. What are those plasmid and their function? Fertility plasmid or F plasmid, this type of plasmid are useful to carry the fertility genes, and they transfer this material from one bacteria to another. Let me take one example. Suppose one bacteria, it can cause a disease to an animal. Another bacteria does not have that capacity to develop the disease in animal. Then bacteria 1 can transfer the disease causing agent as a genetic material to the bacteria 2, and that will be transferred through a plasmid. So when the bacteria is transferring that disease causing agent from one bacteria to another bacteria through a plasmid, that plasmid is known as fertility plasmid. Next plasmid is the resistance plasmid. Resistance indicates resistance against antibiotics. Resistance against antibiotics. Okay. This sometimes when we give antibiotic to kill the bacteria, bacteria immediately try to learn how to survive against this antibiotic. Their knowledge they develop and they store it in that knowledge into the plasmid. And then that same plasmid give and donate to the other bacteria just to learn that how to survive against that antibiotic. So this is the resistance development. We'll talk about the bacterial resistance obviously in details. We should understand it. So this kind of plasmid is a resistance plasmid. Last one is the coal plasmid. You know, bacteria, they do survive in the animal body, human body, in our stomach, in our body, lots of bacteria available. In your stomach, probably there might be more than 200 different species of bacteria. Do you think that 200 different species of bacteria, they sustain in our stomach in a very pleasant and peaceful way? Answer is no. They do fight, they do quarrel, they do try to supersede the others. So there are always fight between the bacteria that who can sustain and who will die. So what do they do? The bacteria, they can produce one important killing agent that is known as colicin, C-O-L-I-C-I-N. Bacteria can produce a very killing agent that kills the other bacteria. Anything a bacteria develops to kill the other bacteria, that is known as colicin, C-O-L-I-C-I-N. Now this colicin also produced by one kind of gene that is available in the plasmid. Okay. Now that same plasmid, when it can pass through the one bacteria to another bacteria, such plasmid is known as coal plasmid. All right. So my dear, this is the all about the day. Now I have 2-3 minutes time. If anyone wants to raise any questions, you are welcome. Anyone wants to ask any question? No question so far? What does it mean? Am I presenting so nice way that everyone is getting it very clearly or nothing? What should I consider? Come on, come on, come on. There should be some questions from someone, or you are not reading at all. Next class, there will be question answer session. Today I missed. Okay, please be ready. All of you, so far whatever we have discussed, first half an hour we'll use it as a question answer session. The topic will be the history of microbiology, then the structure of the bacteria, whatever we have discussed so far. All right?
Yes, sir.
Good.
Yes, sir.
Next day we'll discuss about this. All right, with that, let me conclude it. Thank you, everyone. We have the attendance 64 people today. I am just recording the list. Okay, see you.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir. Thank you, sir.
Thank you, sir.